I am here to introduce our guest speaker, Dr. Vinod Skaria. I think he needs no introduction to the audience assembled here. My co-workers in the university, Dr. Binoj, Registrar, Dr. Manoj Kumar, Dr. Geeta Govindaraj, deans of faculties, professors of our schools, and faculty and learning members of the medical profession, of the health professionals. Dr. Vinod Skaria is a clinician turned computational biologist. Yes, he was here to give an oration earlier in connection with the centenary celebrations of Dr. G.N. Ramachandran on the last month. It was on 10th of October. His research interests are in understanding the function, the organization and regulation of vertebrate genomes and how genomic variations could potentially impact them. He is the co-founder of the Genomics for Understanding Rare Diseases in India, a network called Guardian, which is one of the largest networks of clinicians and researchers in India working on rare genetic diseases. His research has contributed to a number of insights and technologies which are widely being used in India for diagnosis and screening of genetic diseases. I am very happy to state that he had his undergraduate medical education from Kerala, from Calicut University, Calicut Medical College, and his PhD in computational biology from University of Pune. Vinod has over 2,000 research publications in international peer-reviewed journals and 2 books to his credit. He has also been on the editorial board of a number of journals, including Frontiers in Systems Biology, Journal of Orthopedics, Elsevier, Translational Genomics, and International Journal of Rheumatic Diseases. He is a recipient of several awards: CSIR Young Scientist Award for Biological Sciences in 2012, Kavli Frontiers of Science Fellow of US National Academy of Science in 2015, and an elected fellow of the Royal Society of Biology in 2017 and Royal Society of Public Health in 2022. These are some of his achievements. I'm not going to enumerate all of them because there are a lot of such achievements. With great pleasure, I am introducing today's guest speaker, Dr. Vinod Skaria, Principal Scientist, CSIR Institute of Genomics and Integrative Biology, New Delhi, a prestigious scientist from India, for his speech on personal genomes to populations and back. I'm sure that all of you will enjoy it, and we will have a very fruitful deliberations after his speech. Over to you, Dr. Vinod Skaria.
Thank you very much, and thank you very much, Dr. Vijayan, for the quite extensive introduction. I think my screen is visible.
Visible. Very well.
Yeah, thank you very much. So over the next 30 odd minutes or 40 odd minutes, I will briefly describe our journey from personal genomes, that is starting from one genome in the country, to looking at population scale genomes and delving back into helping individuals through whole genome or whole exome sequence. So I have a subtitle, so which essentially says what the journey is all about, on what we have learned from all that we do and why it should matter not just to us, but to people around us, patients around us, and of course, clinicians and healthcare workers and healthcare professionals like you who are in the audience. Before I start my talk, there are a few mandatory disclosures to make because we do work with a large number of organizations within the government and also outside the government to enable genomics and its outreach within the population. Now, the basic premise of what I'm going to start is dated sometime in 2003. And to put in the timeline and facts, this is also the time when, incidentally, I finished my MBBS at Calicut Medical College. And around the world, this was a quite exciting period of time because after decades of very hard work, which involved thousands of scientists, multiple countries and laboratories from across the world, we finally had what we know as the draft human genome. Now, that is essentially the basic code of a human being in nucleotides, A, T, G, and C, and 3 billion of such A, T, Gs, and C organized into chromosomes, which essentially gives the background or the basis, or the molecular basis rather, of what we are. Now, this was very exciting, but this is also very saddening in some aspects, because in 2003, while this was a great human feat, which involved multiple countries, India was never part of the Human Genome Program. Of course, there are many reasons why we were not part of the Human Genome Program. It was largely because we could not put the bill or pay the cost which was required to contribute to this global initiative, to be part of this initiative. But nevertheless, that doesn't matter, and it is very important to also state that the human genome and the data from the human genome is a global wealth, and the fruits of the human genome is for everybody across the globe. Now, the human genome, of course, was not a unique or an isolated event. It happened, the draft was announced in 2003, but at that point of time, there were a large number of other genome programs which has happened in the past and were also happening at that period in time. The genomes which happened in the past include a variety of other organisms, including many bacteria, like, for example, the E. coli, Haemophilus influenzae, yeast, and even, for example, plants like Arabidopsis thaliana, and even fruit flies like Drosophila. And there are far more large number of programs that have happened during that period and after that period, which also took shape anticipating that the human genome sequence would be available. But the real revolution is not really in the number of genomes or the programs that were out there, but largely the technological advancements which happened at the half decade following the human genome sequencing, which would mean that the cost of genome sequencing or the cost of nucleotide sequencing rather would fall very, very drastically over the coming decades. And this is largely possible because of newer sets of technologies, what we today call as next generation sequencing technologies, and clubbed with an extensive array of computational tools which could today assemble genomes in small labs, which doesn't require supercomputers, multiple countries, or $3 billion. Now, we were probably in the right time and the right space because the cost of genome sequencing has been dwindling very rapidly, and it was now affordable for India to actually sequence a whole human genome. And that was possible in 2008 because the cost has become affordable that small laboratories like ours, which are not extensively funded like the Western laboratories, could think and anticipate that they could start sequencing human genomes. So in 2008, we looked at why we should sequence the human genome. And that was largely because there has been an inequity in the distribution of human genomes that were sequenced until that point of time. Now, the top panel that you see here is essentially the admixture plot of the world, right from Africa to America. And each line of this is an individual. If that individual has multiple colors, that necessarily means that the individual has parts of their genome which was admixed, or rather assimilated from a very different population by recombination. Now, at that point of time in 2008, there has been 6 genomes, which includes the reference genome, which was of African ancestry, the Craig Venter and the Jim Watson genome, both of them of European ancestry, and the Chinese, Japanese, and the Korean genomes, all 3 of them from East Asian ancestry. And from the figure, you would figure that all these 3 or 6 genomes that are available at point of time came from quite less admixed populations across the globe. And what you see here in the middle of this plot is essentially an extensively admixed or diverse group of populations, extending from the Middle East to the Central and Southeast Asia. And India is right here in the middle of this large group, which is extensively admixed, today encompasses a third of the global population, but nevertheless did not have a genome representing them. Now, put in context, the Chinese had been part of the Global Human Genome Initiative, and Chinese laboratories have contributed close to around 1% of the human dark genome. But as none of the countries in this part of the world has been part of the Human Genome Program, we never had the resources, the knowledge, and the methodology to be able to sequence genomes. And nevertheless, that was an opportunity, because sequencing genomes from this part of the world could contribute quite immensely to understanding human populations and human diseases. So in 2008 and 2009, with my colleague Dr. Sridhar Sivasubbu, we started putting together one of the methodologies and approaches to sequence the genome using a next generation sequencer. But more importantly, to put the sequences in terms of genetic variants and analyze this genetic variants in the context of what is clinically important or that is functionally relevant. So that is essentially learning the tricks of the trade, because nobody would teach us how to sequence human genomes. You would need to learn it all by yourself by making trial and error. But nevertheless, over a couple of years, we could actually learn these tricks, first starting with the zebrafish genome in 2008, and much later being able to sequence the human genome in 2009. And in the end of 2009, the 8th of December, this was announced in the Parliament of India that India could sequence a whole human genome, making it the 7th country with the capability to sequence whole human genomes. And this is largely possible with a fantastic collaborator, Dr. Sridhar, and 2 young graduate students, Ashok and Ramya, who actually put together the genome and the informatics to be able to assemble the human genome. Now, it's really not the question whether you can sequence genomes anymore, because once you have learned how to sequence the genomes, you could do much more. And that's essentially what we did from the 2009 human genome from India. We later did the Sri Lankan genome, the first genome from Sri Lanka in 2010, the first genome from Malaysia in 2011, and the same year, the first genome from Philippines. And we are also part of the Pan-Asian Population Genomics Initiative, the flagship program of the Human Genome Organization to look at genetic diversity of Southeast Asia. And today in our lab, we do host quite large data sets of Southeast Asian genomes, of the Middle East genomes, and much recently of the Indian genomes from the 1,000-odd genomes that has been sequenced from India quite recently in 2019. I'll talk about these genomes in quite great detail in the coming slides. So the question is not whether we can sequence genomes, but more, can we understand these genomes? Can we make sense out of these genomes? And can we mine these genomes to make useful information that could help patients and families? And now when we talk about genomes and genetic variants, one thing that we need to keep in mind is that each variant, depending upon the variant frequency and depending upon the effect of that particular variant or the consequence of that particular variant, could cause a variety of diseases that we otherwise encounter in normal clinical settings. On one end of the spectrum, you have diseases which we call as common diseases, which are associated with common variants. Now, common variants do not cause such diseases. They only have a very small effect size or a very small contribution to this disease. The commonest examples are diseases that are very common, like diabetes or hypertension. They are not really Mendelian genetic disorders. They're very small contribution from the genetic component. On the other end of the spectrum, you have diseases which are rare in occurrences, but are caused by genetic variants, which have very high effects or very high functionality. And therefore, you could predict an individual just looking at the presence or absence of the genetic variant, whether the individual could develop a particular genetic disease. The best examples for such diseases are, for example, sickle cell anemia or thalassemia or hemophilia for that matter, where the presence of a particular genetic variant in a particular zygosity, whether it's 1 copy or 2 copy, could actually predict whether the individual can have the disease. But nevertheless, we cannot predict whether an individual will have diabetes or cardiovascular disease or hypertension or one of the commonest diseases, because the contribution of these genetic variations to causing the disease is extremely low. So the ways and means to look at such diseases and such variants are also quite distinctly different from each other. On one end of the spectrum, we use an approach called the genome-wide association studies for looking at genetic variations which are associated with common diseases. And on the other end of the spectrum, we use sequencing, whether it's of a gene, whether it's but a part of a gene, it's part of the genome or a whole genome, to look at diseases which are extremely rare, which are caused by variants which are of extremely large odds of causing the disease, but nevertheless, they're very infrequent in the population. Now, while we say that these are rare genetic diseases, the point that you need to keep in mind is that there are around 7,000 such diseases which are documented in literature. And put together, these 7,000 odd diseases collectively affect 70 million people in India and approximately 300 million people worldwide. Now, put 70 million of people in context, is approximately double the size of the population in Kerala who are affected with genetic diseases. Many of them would not even have a diagnosis, would not even know that they have a genetic disease, would go through multiple rounds of misdiagnosis and a considerable amount of time before even they have been diagnosed. Now, put some statistics in here that on average, a rare disease patient goes to multiple clinicians, multiple rounds of misdiagnosis, and multiple years are lost to diagnosis, while also keep in mind that not all rare diseases are treatable, but nevertheless, around 5% today of the genetic diseases which are prevalent in the population have very effective treatments available. And if you can diagnose them very early on, you could have a perfectly normal life. So you think that these genetic diseases are rare, but of course, these diseases are not so rare. And that was a stark recognition that we had on the day we announced the genome in December 2009. Sorry. And on that December night, there was a patient who himself identified as a Bhai, who called Dr. Sridhar, my colleague, and stated that I have read from news that you have been able to sequence the genome in India, and I think that this is a technology that can help me. And just keep in mind, this Bhai was not a doctor, was not even well educated. He was a normal businessman who has a disease or an illness, but nevertheless was intelligent enough to understand that the technology, what we call as genome sequencing, could actually impact him and his family. Now, this individual actually had a very peculiar illness. He was otherwise perfectly normal and a businessman, with a flourishing small business. But nevertheless, whenever he has pressure applied on the skin would break up as if they had burns, and eventually they would form wounds which become infected, which become disfiguring and often painful. Now, this individual was not alone. He had another 28 individuals in the extended family of 4 to 5 generations. And for that matter, he almost knew every one single individual in this extended family of 5 generations, that he could now trace and understand from his intelligence that this is a disease which has a genetic origin, which runs in family, has a 50% chance of inheritance, and nevertheless, genome sequencing can probably provide help to identifying this condition and provide help to prevent the disease from occurring in the family. And this patient was indeed quite visionary. Over the next few years, we could collect all of these 28 affected individuals. We did sequencing of a few individuals. We did identify the genetic mutation. We did confirm this genetic disease as epidermolysis bullosa simplex. We published this particular paper suggesting this is the largest family of epidermolysis bullosa simplex documented in global literature. And more importantly, apart from all of these, the identification of the genetic mutation enabled the family to do genetic counseling, that every single individual who's in the younger generation, who is prospective for marriage, could test for this particular genetic disease. And more importantly, you could do prenatal testing to prevent the disease from recurring in the family. And today, a decade later, you could say that genetic testing has practically eliminated the recurrence of the disease from happening in this particular family. And that is really the impact of normal people with very good intelligence who can really understand the impact of technology, not just in helping themselves, not just in helping their family, but also helping society at large. And if you think that these are isolated cases, that is really not true. And in many ways, India has a very large advantage, being a very large population of 1 billion people. That is approximately a sixth of the world population today. And unlike many of the large populations, we have a very diverse population with very different genetic ancestries. It is quite stratified by endogamous groups. Endogamous groups essentially mean that they're populations who marry only within themselves by their surnames typically, or by the caste or creed. And there are around 4,600 such endogamous, well-defined groups in the country. And more importantly, we still have a close-knit family structure. And as I stated before, we could draw pedigrees which could range 3, not 4, not 5 generations, which is really impossible in many other countries which has been displaced by multiple rounds of world wars, multiple rounds of famines, multiple rounds of migration. But nevertheless, India as a population has largely remained in situ without much of a disturbance for centuries altogether. And all that we do in genetics starts with an extended pedigree. And fortunately, we can extend a pedigree for, if not 3 generations of individuals, but probably many generations. If the highest number of generations that we could trace is one family for around 54 generations. So in 2015, with Dr. Sridhar Sivasabu, we started this program called Guardian. Guardian stands for Genomics for Understanding Rare Diseases, India Alliance Network. The idea and concept of this program was quite simple, that we would work with clinicians who would see patients in their clinics, in their hospitals, in medical colleges. And if they have a patient with an undiagnosed genetic condition, but nevertheless have a familial recurrence, or there are multiple individuals in the family, they could readily refer these families to us. We would consent, take a 5 ml blood sample, sequence their genomes in an attempt to identify what is really wrong in the genome of this individual. And whenever we find a genetic mutation, which can be assigned as a pathogenic genetic mutation, we would return it back to the clinician. We would tell the clinician that we have found this particular genetic mutation, and the clinician could act upon that particular information in terms of providing genetic counseling to the family, in terms of offering additional screening to the extended family, in terms of offering prenatal testing, and also advising the family on how to eliminate the disease in the next generation. But that is possible in only a subset of individuals, because even with our best of our efforts, the positivity or the chance that we would identify genetic mutation from the referred samples is still quite low. It's approximately around 50%, but quite variable across different specialties. So we would need to do quite extensive analysis, including extended family pedigrees, a lot of wet lab work, which might actually take years altogether before we can actually prove that a particular genetic mutation that we found was indeed what caused the disease. And that's essentially the science that we could do on this population scale samples. And whenever we find a genetic mutation, we could get back to the clinician and tell that we have found this genetic mutation. We have all evidence to suggest that this is pathogenic, and therefore we could act upon it. And from 2015 onwards, this program has been exclusively funded by CSIR through multiple rounds and multiple grants. And today it has grown to encompass close to around 80 clinical centers across the country, and encompassing 300 clinical collaborators directly and thousands of other clinical collaborators in an indirect way. So to give you examples of what we do, we build collaborations at a scale which was never seen before in the country, in doing research for rare genetic mutations, educating clinicians and extended members in the community, in offering genetic diagnosis to a subset of rare genetic conditions, in terms of implementation of technology for rare genetic diseases, and influencing policy for rare genetic diseases in the country. For people who are interested, there is this extensive review of this article published in 2020, which has all the 280 authors at that point of time from 70 different centers across the country. To give you examples, and this is just a few examples on how and why this could happen and what's the impact of what we do. Now, this is one such family which was in newspaper way back in 2015. And with a curious case that they had petitioned the President of India to euthanize 6 of their 8 kids. Now, think of the plight of the family who would petition to euthanize their own kids. And they did this out of frustration and out of hopelessness that they had a genetic disease which runs in their family. Children are typically born normal until they are around 3 to 4 years of age. And at that point of time, they progressively deteriorate in their milestones, and eventually they end up in a vegetative state and they die. And despite multiple attempts to find what is really wrong with these kids, there was no diagnosis, and therefore the family petitioned the president to permit them to euthanize all 6 of their kids out of their 8 kids. Now, this family, of course, was referred to the governmental channels to us. We could do an extensive genetic study of this particular family. We did identify the genetic mutation which caused this particular disease, and also give the diagnosis as megalencephalic leukoencephalopathy with subcortical cyst type 1, an extensive name for a disease, but in short, what is called as cystic megalencephaly, caused by a genetic mutation in a gene called MLC1. Now, this is an extensively rare genetic disorder, or extremely rare genetic disorder, and this particular mutation was previously described only once in a family from Persia, and just one individual in that particular genetic report. Now, of course, this was very curious to us because if the original individual which was described quite long back was from Persia, what has this family to do with that Persian individual? And through extensive home-to-home surveys, we did identify that almost all these individuals did belong to a particular community called the Nalband community. They trace their origin from the Middle East. They came with one of the invaders and they settled around Agra because they had the art of making horseshoes. Now, the extended door-to-door survey also identified another 3 families with the same genetic disorder and also tested positive for the same genetic disorder. And more importantly, screening individuals in that particular community, we did realize that approximately 28% of individuals in that particular consanguineous community were carriers for this extremely rare genetic mutation. And this is what we call as a founder event. Or in other words, a founder individual or set of individuals who had this particular genetic mutation formed progenies in the community who eventually married within themselves because they have a consanguineous marriage practice and therefore could increase the frequency of a particular genetic disease in a particular population, otherwise rare in the rest of the world. So from the first case reference 2016 to completing the diagnosis in 2017 to publication in 2018, we today have a diagnostic in the market which tests for the MLC1 genetic mutation in the Nalband community, which today could impact 50,000 odd individuals from this particular community. We also have this particular test for another founder mutation for the same disease, but nevertheless in the Bansal Agarwal community. And therefore, these 2 genetic mutations, which are just for 2 founder mutations in the Indian population, could impact a very large number of individuals in the community. And this entire work was done in collaboration with Professor Shefali Gulati from All Institute of Medical Sciences and a charity organization called the Human Welfare Foundation, which works among the community members, the Nalband community. Now, of course, there's another such family and now another such individual. This is a 30 to 29-year-old businessman, very well educated, was an engineer by education. But nevertheless, he went to multiple clinical centers with fever, which recurs every 3 to 4 months. He has been diagnosed and tested for every possible infection, including tuberculosis. He has been put on multiple rounds of anti-tuberculosis treatment regimen, but nevertheless, the fever recurs, and every time there was no culture, there was nothing that would be found as positive that could suggest an infected origin. The extended pedigree of this particular family suggested that this was a quite consanguineous community. And by extensive genomic analysis, we did suggest that this individual has a particular genetic mutation in the MEFV gene, suggesting this is a disease called familial Mediterranean fever. The familial Mediterranean fever belongs to a class of disorders called periodic fever syndrome. Or in other words, these individuals would have recurrent fever, which sort of mimics infection in a recurrent way, but nevertheless, this is not caused by pathogens, but probably the body overreacting to itself, thinking that there was a pathogenic attack. Now, familial Mediterranean fever is a disease which is very prevalent in the Middle East, as the name suggests. So it was very interesting to also prove that this individual did indeed have a Middle Eastern ancestry. And to put things into context, you could use genomic tools to not just identify the genetic mutation, but also confirm the ancestry of the individual by showing that this individual had a chromosome or part of the chromosome which traced back to the Middle East by many generations. Or this is what is called as an admixture analysis, which could tell what part of the chromosome did come from what part of the world. And this was actually published much later, suggesting the individual had an Egyptian ancestry by around 54 generations ago. Now, if you think that these are isolated cases, these are not. There are a large number of populations today in India. They're in all aspects Indians. They came as friends, they came as businessmen, they came as traders, much later they came as invaders, but nevertheless, they've always remained Indians in all aspects of culture, all aspects of tradition, but nevertheless, they have a small portion of the genome which traces their ancestry to a different part of the world. And when they trace their ancestry to a different part of the world, they also have diseases which are prevalent in a different part of the world, and therefore they remain undiagnosed or underdiagnosed because these diseases are not very prevalent in this part of the world. And therefore, it's very important to also look at the ancestry of the individual and or put the disease in the context of the ancestry to also make very useful guesses. And not just that, it's also very important to collaborate quite extensively with global populations elsewhere. This is just a small snapshot of people who trace their ancestry to the Middle East, but there is a large number of populations from across the world who have come to India, or Indians who trace their ancestry to different parts of the world. And even more importantly, there's a quite large Indian diaspora who trace their ancestry back to India, but nevertheless today are across the globe. So that is essentially why genomics is therefore a global enterprise, and why it should be very important to collaborate, not just to identify genetic diseases in the country, but also genetic diseases in context of where did they come from and where did they arise from. Now, of course, there are many such conditions where we might not be able to identify genetic condition on the first attempt. And this is one such case. This is a family who was tested and referred to us from the All India Institute of Medical Sciences, Dr. Rajneesh Juneja, suggesting a mitochondrial genetic disorder. We did sequence the mitochondria. A mitochondria is a 16,000 base pair organelle and which is separate from the nuclear genome. We did sequence the mitochondria. We did not find any mutation. We further sequenced the exome. We did not find any mutation. And years later, when we could sequence whole genomes, we could find a large deletion in that particular individual. And more importantly, by identifying this genetic mutation, it's very important to now prove that this genetic mutation caused the disease. Not all genetic deletions would cause the disease. So you need to actually put that particular genetic deletion in a model system, and the model system that we use is zebrafish, or in this particular case, also cells derived from the patient. And it takes quite some amount of time to actually go back and prove in the lab that that particular new deletion that we identified indeed caused the disease. And this particular system took around 9 months, which is a very good thing to do. And what we could prove is that this individual did indeed have a mitochondrial disorder, but the genetic deletion was in the nuclear genome for a very important gene that was translocated to the mitochondria or was important for mitochondrial function. By looking at the cell lines of the individual, by looking at a zebrafish which has exact same deletion, we could prove this genetic mutation as pathogenic. We could go back to the family with a very cost-effective test, which is based on MLPA, to be able to query this genetic deletion and do screening in the community, and of course do prenatal testing to prevent genetic diseases from recurring in the population. And then comes to the real question on what is the future going to look like. There have been a number of genetic tests that people have been using. Quite extensively, people have been using gene sequencing, people have been using gene panels, people have been using genetic whole exome sequencing. But nevertheless, this point is to make, what I'm trying to make the point is that the future belongs to whole genome sequencing. And there are multiple reasons why we think so. The first reason is that it is extremely fast. You can actually sequence a whole genome in less than 24 hours. And more importantly, a whole genome encompasses all the 3.3 billion alphabets in the genome, and not just part of it, and therefore it is all genetic tests put together. So you don't need to do a test for a chromosomal deletion, you don't need to do a test for a chromosomal translocation, you don't need to put a test for a single nucleotide variation, you don't need to put another test for a repeat expansion. You can encompass all with one single test. And increasingly, the cost of whole genome sequencing is reducing very rapidly, and it's assumed that probably in a year or two, we would have whole genomes at the cost of whole exomes. And that would have immediate applications in a variety of clinical applications, including neonatalized use for sick neonates, quick diagnosis for familial genetic conditions, quick prenatal screening where time is critical. For example, there is an undiagnosed sibling birth with a rare chromosomal abnormality which has not been tested. So quick turnaround time, it is possible to actually screen individuals and identify the genetic. Now, all of this comes to the very basic premise that how do we know what is prevalent, or what do we know as prevalent genetic disease? Now, coming to that point of prevalence, there is always a big conflict. And the conflict is that if you ask any clinician, the clinician will tell what's prevalent that he has diagnosed or what he thinks is what the disease he has diagnosed. Now, assume that a basic premise to start with is that a clinician do not diagnose such conditions, right? Which are extremely rare genetic conditions that normal clinicians do not diagnose. So if that was the case, how do we really identify a prevalent genetic disorder? And that is today possible because you can ask a question the other way around. You can look at normal individuals in the population and ask a very simple question: what are the genetic mutations that are carried by normal individuals? And if they carry an abnormal mutation at very high frequency or a pathogenic genetic mutation at very high frequency, the disease is very prevalent. And if the disease is very prevalent and the clinicians are not diagnosing them, then we need to go back and educate the clinicians so that they can diagnose them. So in other words, we should not really rely upon the prevalence of diseases coming from hospitals, coming from clinicians, because the very basic premise is that clinicians cannot diagnose extremely rare conditions on a routine basis. I'm not saying all clinicians cannot identify. They're very specialist clinicians who can identify very specific diseases because they're trained in it. So the question is, can we train such clinicians to identify undiagnosed prevalent conditions? And for that matter, the really important type of prevalent conditions which are treatable so that we can afford an early diagnosis so that patients can have a perfectly normal life. Now, as we talk about genomics and analysis of genomes, there's also a drastic change in paradigm, because typically what we talk about genomics or clinical genomics is that the patient is referred by the clinician and the clinician therefore advises a genetic test. And the genetic test is advised with suspicion for a particular disease or a type of disease. And the report comes back to the clinician saying that this particular disease exists. But now, what if we could sequence a newborn at birth? Of course, the newborn might not have a genetic condition or a phenotype to start with. The disease might develop a few months later, a few years later, we don't really know. Can we figure out that the child indeed has a genetic mutation which would put the child at risk for a genetic condition? And for that matter, you need to have evidence for the genetic variations and evidence for actionability so that you can go back to the clinician and tell that this normal looking child probably is at risk of developing a genetic condition and therefore warrants clinical intervention. That is really the paradigm shift that is happening today, and that's essentially what's going to drive genomics in the future. And to do this, what we need to understand is what is really prevalent in the population. And to understand what is prevalent in the population, you need to sample individuals from the population and ask a very simple question: what are the pathogenic genetic mutations in the population? And from this prevalent pathogenic genetic mutations, can we go back to clinicians, give them heads up and say that these are the kinds of diseases what we should identify. These are the symptoms for these diseases that we should identify, so that we can equip clinicians to identify them very early on so that they can be acted upon. So in 2019, CSR funded us with this program called the Indigen, which was to do the first pilot 1,000 individuals from the Indian population. So each dot out here in the map is essentially where the individuals have come from. These are apparently healthy, normal individuals. Now, what do you do with this? Of course, the numbers are very small, 1,000 or a billion population is extremely small. That would mean that we have sampled 1 in every million individuals. Of course, we need to do more. With more money and more funds, we could probably do more. But nevertheless, with the funds that we could afford, we could do 1,000. So we could look for treatable genetic disorders which are prevalent in the population. We could look for adverse drug reactions which are prevalent in the population. We could look for cancer susceptibility variations. We could identify population specificity for genetic diseases. And the basis for all of this, as I said before, is that you need to identify genetic variations which have evidence to be saying that these are pathogenic. And to do that, you need to actually create quite extensive databases. And this is one such database that we have recently created for hereditary breast and ovarian cancer for the BRCA1 and BRCA2 genes. So essentially what we do is we collect every single genetic variation that has been described in literature, go back and systematically reevaluate evidence on every single variant and put them into the context of whether they are a pathogenic variation or they are a benign variation. And based on the evidence, if they're a pathogenic genetic variation, you could now use those variants to look at the frequency of those variations in different populations and come back and say that this population is at a higher risk or this individual is at a higher risk of developing hereditary breast and ovarian cancer. And you could do this for n number of diseases today. And more importantly, as we do this and as the evidence changes, it's very important to communicate this back to the individual. So we have piloted a program which is called the Indigen Card, which is essentially a digital service which will provide the evidence on the genome, not the genome. It doesn't access the genome. It provides the evidence on the genome in a secure, privacy protected point of care, and more importantly, clinician enabled fashion. So every single volunteer who participated in the Indigen program has a card and an app, like what you see here. And on this app, the clinician can request for the genetic analysis, or rather, the evidence which is available for clinical actionability on that particular individual. And it's not really the genome that is being described, but the evidence that is overlapped to the clinical genome that is available to this individual. And by doing this, of course, you could identify prevalent genetic diseases. Dr. Geeta Govindaraj is in the audience. Over the last 4 years, we have created a quite elaborate program for primary immunodeficiency disorders with the help from the government of Kerala, with the help from SERB, grant, with the help from a charitable organization called the FPID. We could identify genetic diseases called the primary immunodeficiency disorders, take it to diagnosis, take it to policy for treatment and for hematopoietic stem cell transplant. And more importantly, with extensive clinical collaboration, take it to community interventions that the community could be educated and screened for prevalent genetic conditions. And today, Dr. Geeta Govindaraj runs a specialized PAD clinic in Calicut Medical College, which has benefited a very large number of patients who would and who are today alive because of the fact that these disorders are identified very early on. They have been put on treatment, IVIG, for subset of individuals, and hematopoietic stem cell transplant for another subset of individuals, all supported by the government of Kerala. And today we identified some diseases which are prevalent, some mutations which are prevalent in the population, so that we can offer very targeted, sequence-specific or disease-specific diagnostics which are implemented at Calicut Medical College. So you could find many such examples from the Guardian program, where from single individuals to populations to communities, you could impact patients and families. And this is just one medical college. And think of the impact where you can extend such a study to across medical colleges in Kerala. So for example, all the medical colleges in Kerala are under the KUHS. And this is one such example where you can today use evidence which is already available to extend such an understanding to colleges across Kerala. And this could significantly impact a large number of patients who remain undiagnosed, who die undiagnosed, but with the right diagnosis, with the right treatment, today they can live a perfectly normal life. There are many such examples. There's another such example from the genome sequence. We identified a prevalent genetic mutation, a pentanucleotide extension repeat. We went back, identified families and communities, and today we have an extensive large cohort of around 400 odd patients. And to give context for this particular genetic disorder, which was not even identified until 2018, and there are only 32 reports for this particular genetic report, 32 individuals reported for this particular genetic disease. Today we have 434 molecularly characterized individuals out of just one medical college in India. And today we have a very cost-effective screening test using RT-PCR, which could today impact the community size of 5 lakh individuals. If COVID did not strike, today we would have a much larger cohort of individuals. And more importantly, diagnosis of this disease could enable them to access treatment with a very cost-effective treatment modality available. Now, of course, the priority of any country should be to prevent diseases which are diagnosed at a newborn stage. And that points to the question, what should we really screen for a newborn? And of course, we cannot yet do whole genome sequencing for every newborn in the country. We are not that rich. There are some countries which are rich. But of course, that doesn't mean that we should wait or our kids should wait till whole genome sequencing becomes cheap. So the real question is, what are the prevalent genetic diseases? For example, one use case that we have taken is treatable causes of intellectual disability. There are around 104 genes, mutations, or monogenic disorders. And you could go back to the indigenous data set and ask a very simple question. What's the prevalent pathogenic mutations in these 104 genes? Quite extensive job done by a student, Aditi Batra, over many years. Today, we have a count of what is prevalent in the population. We have diseases like phenylketonuria and Wilson's disease. It's as frequent as 1 in every 166 individuals. We have diseases like citrullinemia, which is prevalent 1 in 200 odd individuals. In other words, these are probably the prime candidates that we should actually take up for newborn screening in the population. For Wilson's disease, of course, today we have taken it much further ahead. This is work by Dr. Vinukumar PK, a clinical collaborator of ours and an extensive colleague and a friend of mine. And over the last 4 years, he has created a very formidable resource integrating clinical information for Wilson's disease, creating one of the largest cohorts for Wilson's disease in this part of the world. And not just creating the cohort, he has also created a very cost-effective genetic diagnostic, which today can screen to 100 to 3,000 samples in a day in a very cost-effective configuration and amenable to today newborn screening. He has also created world's largest sets of evidence repositories for Wilson's disease and also integrated AI and ML approaches to identify genetic variations and prioritize genetic variations for further study. So the idea is that we today understand quite a bit more about what are the prevalent genetic diseases, and we do have tools and methods to tackle such genetic diseases, provided we could work very closely with a wide variety of individuals. And to give another example, this is another such disease. 1 in every 200 individuals in the community is suffering from this disease called familial hypercholesterolemia. Now, how do you diagnose familial hypercholesterolemia? Any individual more than 190 milligrams for LDL should actually get evaluated for familial hypercholesterolemia. Today, every biochemistry lab does this particular test, and there are n number of individuals with this cutoff who are still walking around without a diagnosis of FH. How do we make this difference? We could make this difference by educating clinicians that everybody above this particular cutoff should be looked for very closely for tendon nodules, for xanthelasma, for arcus cornealis, and for a family history of early cardiovascular events. And if you could do this, today we have a very cost-effective test which pretty much costs as much as testing LDL cholesterol. That you can make a molecular diagnosis. With a molecular diagnosis, they can be put on statins, and with statins, they can have a quite normal life. So in summary, what we have been trying to do is to essentially work with clinicians to understand genetic diseases today through the network what we call as Guardian. We also offer very specific tests of prevalent genetic diseases through a program called COMED, which is done by my colleague Dr. Mohammed Farooq. And in this process, we educate a large number of clinicians to multiple different programs. And we have instituted the first genetics fellowship program in the country with Sanofi Genzyme, and much recently an online program called the Genomic Variant Analysis and Clinical Interpretation over the last 3 years, reaching out to over 5,000 clinicians, not just in India, but from 40 different countries across the globe. And by doing this over and over again, we could also add value to the diagnostic industry and also make them build and execute this test in a very cost-effective fashion, so that a very large number of people who are not within our network or not within the clinician network that we work with could access such genetic tests. And more importantly, it's important to also think about a permanent cure for many of these conditions. And that's where we work in the lab to look at multiple different approaches, molecular, including genome editing and cell-based therapies, with multiple different colleagues at IGIT to find cures for prevalent genetic conditions. I think I'll stop there and take questions. But before that, let me say that genomics can make medicine extremely predictive. It can predict an individual who is likely to or at risk of developing a disease. It can prevent genetic diseases from recurring in communities and families. It can make a diagnosis extremely precise or molecular, and treatment personalized. But the centerpiece is more important, that participation, not just of clinicians, not just of patients, but also a lot of people around them, including patient support groups, including researchers and students around us. I think I'll stop there and take questions, but before that, let me acknowledge a lot of people who made this possible. Fantastic set of clinical collaborators, many of who are also in the audience. A fantastic set of students who have been in the lab in the past almost for a decade, who contributed to multiple different programs and cohorts. Collaborators from across the world who helped us understand genomics much better. And of course, this happens with funding, and funding has consistently come from CSIR to multiple different grants over the last decade and a half. I thank you, and I'll take questions. But before that, for clinicians who are interested in learning more, we have an online course which you can register for free. Thank you very much. I could take questions if there are.
Thank you, Dr. Vinod Skaria, for the impressive talk on this personal genomics. During our undergraduate years, we thought that the genetic diseases is a problem only among endogamous groups. And now we know that there is a hidden burden of genetic diseases in all communities. And the genome sequencing will lead to a diagnosis of several diseases which are going undiagnosed. And we are happy that our Dr. Geeta Govindraj from Calicut is involved in genetic diseases, a study on genetic diseases. And she was planning to have a course on genetic counseling. The initial thoughts are on the way. It is in the pipeline only. After hearing your talk, I'm sure that the relevance of such a course is more and more. And I'm very sure that Geeta may be having, Geeta will be talking on it. And this, I'm happy to announce that the topic is open for discussion. Dr. Geeta, please.
Okay. Thank you so much, sir, for your kind words, and thanks, Vinod, for that wonderful presentation. As usual, we expected nothing less, and it has been an absolute pleasure. And we have learned so much, and it's an uphill curve. There's so much more to learn, and we are still just at the beginning. But we are so happy that we have been able to help so many families. And after joining KUHS, I'm sure with the proactive environment and all the leaders we have, we would be able to do further work, both in terms of training as well as research. And as Professor Vijayan already has mentioned, we are in the process of planning a course. We would like to have a hybrid course, that is online as well as contact sessions. And MSc in genetic counseling is what we are working towards, and we hope it will materialize. And I think KUHS should step in and make this the meeting point between the clinicians and the basic scientists. It doesn't happen most of the time. And I think KUHS is well placed to bring basic scientists and clinicians together so that they could discuss on what problems and undiagnosed cases and the problems that are unsolved for several years. Things can definitely happen when this liaison between clinicians and basic scientists happens. So I hope that is something that will happen in KUHS. Another thing I also hope is that newborn screening will one day become possible for severe inborn errors of immunity, including severe combined immune deficiency. And that is also something that we are looking forward to. And I would like to thank again Dr. Vinod and his team for the wonderful collaboration that we have. And I hope we'll be able to take the work forward in the coming years to come. Thank you.
Let us hear from the participants now. Yes, I think there are questions in the chat box. Dr. Vinod?
Yes, sure. Okay, the first question is from Dr. Sebin Kumar. I'm a psychiatrist. Is there any advances in genomics in Canada that I can make use of? Okay, it's a very interesting question. Now, what we have realized is when we talk about very prevalent treatable disorders, right? For example, that I gave as the work of my colleague, Dr. Vinu Kumar. Now, Wilson's disease is a very prevalent disease. We know from the genomic data that it is one of the most prevalent recessive treatable intellectual disability disorders. Early diagnosis could make a difference. And there's a textbook example of what a Wilson's disease patient would look like. And typically, they're taught in pediatrics, and pediatricians think that they would see all Wilsons. Now, increasingly, what we are seeing is that Wilson's could also, in some individuals, present much late on as psychiatric disorders, and incidentally found on close examination. And unfortunately, a psychiatrist is not the person who would examine a patient otherwise. Are the kind of people who rather talk to the patient rather than examine the patient very closely. And if you really go back to the literature of Wilson's disease, you would realize that even the earliest cases of Wilson's disease were actually discovered by psychiatrists and patients who come with psychiatric abnormality rather than the classical Wilson's phenotypes of a child with hepatosplenomegaly and liver failure that would come across. So, of course, there are many disorders many genetic disorders which is present in the psychiatry. This is one such example. There are many more, and of course, we could take that offline as we speak. There's another question. Will you please elaborate the treatment aspect of these diseases and how to prevent these types of diseases in the next generation? And I'm sure Dr. Geeta Govindaraj would be a better person to answer this. in my slide, I sort of mentioned one such genetic disorder, which is very prevalent, which is primary immunodeficiency disorders. It encompasses around 300 gene disorders today, close to around 400 today, gene disorders. Early diagnosis of subsets of disorders, like for example, SCID, could be treated with very effective hematopoietic stem cell transplants. Now, of course, that is something that is expensive, that also needs to have a matching donor, so on and so forth. But nevertheless, it becomes a curative treatment, and patients who have undergone this treatment, of course, with additional risk, could anticipate to have a perfectly normal life. Some subsets of primary deficiency disorders, especially the B-cell abnormalities, could be treated by IVIG, or intravenous immunoglobulins, which today are available and is also supported by government programs so that the patients can afford to have them, and they need to be taking this every 3 weeks to 4 weeks on a regular basis. So there are treatments available. It's not that all treatments are 70 lakh rupees or a crore worth. Many diseases, including primary immunodeficiency disorders, Wilson's disease, hypercholesterolemia, they're all very treatable disorders with very cost-effective, very safe medications, provided we could diagnose them very early on. And it's estimated that approximately 5% of genetic disorders today are treatable, provided we could make the diagnosis very early. There is a question from Dr. Indu. Any genomic advances available regarding squamous cell carcinoma in Kerala population? I'm not really sure because when we talk about cancers, we typically talk about somatic mutations and environmental problems. There are genetic factors like, for example, H3 breast and ovarian cancer, now H3 prostate cancer, so on and so forth, which are familial in occurrence. But I've not really come across any literature in terms of squamous cell carcinoma, in few exceptions, except a few exceptions that they could be familial, but nevertheless, they are not very prevalent in the population. The other question is from Mohandas sir. Many neonatal deaths are undiagnosed or wrongly diagnosed. Can we have a way to get WS done on sick newborns and have diagnosis before they die and think of definitive treatment? Yeah, so the answer is absolutely yes. Technically possible. There are some countries which are not even doing WS, they're actually doing whole genome sequencing because, as I stated, whole genome sequencing is much faster, but nevertheless quite expensive at this moment. So for having every sick newborn to access whole genome sequencing, yes, probably we might need to be a bit more developed country. But until we are at that point of time, we should actually look for prevalent disorders which are prevalent in the population. And that's why I suggested that we could look at priority diseases, for example, treatable conditions, where early diagnosis could make a difference. Primary immunodeficiency is just one example of it. You could have metabolic disorders like Wilson's, citrullinemia, phenylketonuria, so on and so forth. You could look at cardiac channelopathies, which is another large set of disorders which could be identified with just an ECG. You could look at familial hypercholesterolemia, which could be identified with just a biochemical test. So there are a variety of such genetic disorders which are very prevalent in the population, which has a very effective treatment available, and therefore we should prioritize them on first go. But nevertheless, we should also not think that we are poor, therefore we should not do whole genomes. We should do whole genomes for quite needy and or quite what do you call required cases to investigate at this point of time, and also be prepared, more importantly, to a point in time where whole genome sequencing becomes cheaper and effective. And that time is not far away. It's going to probably happen in half a decade to a decade, where you would be able to afford whole genome sequencing as a routine test for sick newborns in the ICU. But more important is not really getting the whole genome sequencing. It will happen. More important is to actually get the skill sets to be able to analyze the genomes, and also create the workforce to be able to handle once a diagnosis arrives. And that's essentially where genetic counseling becomes extremely important, right? The moment you're going to identify 1% of newborns with genetic disorders, you're also going to have to need a very large workforce of individuals who are going to educate the family, help the family through, and also counsel them appropriately. And that is essentially going to be the roadblock, because not many clinicians are aware about genetics and genomics. Not many people in the community are aware about genetics and genomics. And unless we have people who can actually bridge the gap in knowledge and advise accordingly, there's going to be a large gap. We should not have a population which does a lot of genomes, but no impact, because if the patient is not advised appropriately, the patient is not going to benefit from the whole genome sequence. So that's essentially where I think the plan from KUHS to create a program for genetic counseling is extremely important, because we should prepare ourselves when we get a large number of individuals where the genome studies are going to happen. I think that's all questions that I have.
Dr. Anil Kumar has a question. Sir, please introduce yourself and ask it.
Yeah, I'm Dr. Anil Kumar, Professor HOD Psychiatry, Toronto Medical College. So thank you, Vinod Sakaria, for the informative talk, giving insight into the genetics. So regarding mental illness, is there any initiative in India or in your lab regarding, because the bipolar disorder, there is a high genetic loading, schizophrenia, and even ADHD. So there's a lot of stigma carried with the diagnosis.
Right.
So the prediction or the future chance predicting is also carries a risk. That's one thing. And another advantage is, for example, in child, I'm mainly dealing with children, child psychiatry. Children with ADHD may be showing the similar pattern of a manic bipolar disorder. So if we can diagnose this with a manic, with a genetic thing, we can change the treatment pattern. That we are using methylphenidate stimulant for ADHD, which may worsen mania. So it may be a therapeutic advantage also. So what is your opinion on this area?
Right, you're absolutely right that many genetic disorders could present as psychiatric abnormalities, where the underlying cause is a metabolic abnormality, but that is manifested as psychiatric abnormality. In terms of genetic studies, yes, there are multiple genetic studies happening in the country on psychiatric disorders. Of the largest note, what I can think of is a program that is done by NHANES with NCDS, which is called ADPS, which looks at bipolar disorders and schizophrenia. But apart from that, there are also smaller programs which look at Mendelian genetic disorders. And as you would also see, a significant large number of bipolar disorders in schizophrenia would actually reoccur in family, and some of them actually show a Mendelian pattern of inheritance. So such extended families, I'm not saying two individuals in the family, there are families where 3 and 4 generations of individuals are affected. Such extended families, genetic studies could actually provide insights into Mendelian genetic causes for psychiatric disorders. And India is a unique place to do such studies, because you can actually trace families 3 to 4 generations, which is usually impossible in many other parts.
One more clarification. I have read that for ADHD, there are 2 types, like VAL and MET allele. That the methylphenidate response is more in VAL. It is not in MET. So is there, on that facility, is it available in India to make a diagnosis that psychogenome, pharmacogenomics?
Yeah. So you're respond. Right. You're absolutely right. So you're talking about another area which is parallel to Mendelian genetic disorders. It's called pharmacogenomics. And the idea of pharmacogenomics is the fact that every drug that we take is either metabolized or excreted by genes or proteins which are encoded by these genes. And therefore, genetic mutations in this gene could affect how the drug is metabolized, excreted, and how drug acts upon the individual. Now, while widely psychiatric drugs do have pharmacogenomic markers, there are at least a few of them which are in the clinical pharmacogenomic implementation consortium, which is essentially an evidence base which says what variants need to be looked at in what set of patients. They're not widely available, but today there are tests available, and there are companies offering these tests for a wide variety of pharmacogenomic markers. And the problem for pharmacogenomic markers is also that in many cases they have to be preemptively provided before the treatment is given, and that becomes what do you call an educational roadblock, because most clinicians are not aware about the fact that what are the tests that I need to preemptively do before I administer an anti-epileptic or an antipsychotic medication. So the real gap there is education, and fortunately I don't have the slide here, but we do have an online program coming up. You can access in the same link that I provided, which provides an overview of pharmacogenomics as a field to educate clinicians on what drugs would have evidence, what are different specialties which would benefit from pharmacogenomics, and what are the tests which are available and who provides it. Thank you.
Doctor,
Thank you.
Dr. Indu PS has raised her hand.
Thank you, Vijayan sir. Thank you, Vinod, for the excellent discussion. Just to continue the thread of discussion from Dr. Anil Kumar's query in relation to mental health, there is a lot of scope for research because since IGAB and CSIR have both, of course, personally at your level also have been working with viral infections, genomics, COVID, and lots of public health significance. What's the scope of research, whether it be pharmacogenomics or diagnostics, in relation to the university? What's the scope? For example, whether it be postgraduate research, PhD research, of course, KUHS is coming up. There's a lot of emphasis on research. So, what is the tie-up, the collaborative part? What's the scope, IGIB and CSIR in general?
Yeah, so thank you very much for putting that question in. So I will tell my opinion. Don't confuse it with my organization's opinion or anything. It's my personal opinion. So in my personal opinion, what we should look at is think of what's the purpose of KUHS. KUHS is a university which is going to educate the next generation of clinicians, or rather healthcare workers. It's not just clinicians, it's also nurses, it's also paramedical people, but largely the healthcare workforce of Kerala for the next set of generations. Now, being an educational institution, the most and utmost importance is to actually advance the field in the local context. And both of them are important, advancing the field and also the local context. What typically happens in a university is that we would want to do research in a field that doesn't really affect us, right? That is not going to make any difference to anybody. We need to advance science on problems which are prevalent, which are local, and address problems which are going to impact people, because you're going to train the next generation of people. And the best example that I can sort of put together, and I'm sorry, madam, for getting back to you every time, right? Working over the last 5 years in Calicut Medical College on one focused disease. It's not just we don't work on other focused diseases, but this is an example right here in Kerala, right near KUHS, which is also part of KUHS, in fact, where just by starting a program on education and research, right? Less of research, more of education is what I would say, right? Today, we have a large number of PGs who have spread across the state, who today can understand and sort of look at a patient and say that this could probably be primary immunodeficiency, right? Even that small change is going to make a huge difference in diagnosing such patients, referring such patients, and ensuring that they can have a perfectly normal life. I mean, think of it this way. If they were not educated, they would not suspect the case. The children would die without a diagnosis. Today, just by suspicion, just because Dr. Geeta Gondaraj has been going over talking about PID every day to every resident that comes to the department, right? Today, residents are aware about the disease, right? They can actually go understand, read more, and suspect such disease. Now, if you can do this for every prevalent disorder in the state, right, we have made a huge difference, right? So that's point number one. Point number two is that as a university, what we need to keep in mind is that we need to be prepared to the future, right? I sort of explained how the future is going to change and how genomics is going to be one of the many arrays of tests that probably a newborn has to go through. Now, the question is, assume that that happens tomorrow, right? Assume that you have a $10 genome tomorrow. Every kid would be able to afford, and then I'll say government will be able to afford a $10 for every kid for a newborn screen on whole genome sequencing, provided it becomes $10. Assume that it happens tomorrow. Do we have doctors who can analyze the data? Do we have doctors who can interpret the data? Do we have doctors who can communicate that information? Do we have doctors who can say that this is evidence and this is not? this is actionable and this is not? The answer is no, right? How are we going to prepare ourselves to that future, right? That is extremely important being a university. Right? There's no point in learning the tricks after the world has changed, right? The point is to actually make the change and grow in anticipation with the change. And to do this, I mean, to train clinicians in genetic counseling, to train clinicians in genome analysis, it doesn't cost anything, right? You don't need hundreds of crores of rupees to sequence genomes to train these people. No, you will probably require fractions of crores, probably just lakhs, to actually create programs so that clinicians are aware, create pilot programs so that clinicians could be trained, and to just put in numbers. Dr. Geeta Govindaraj will say that how much of the research grant, right, ran this program for 5 years, right? That is pretty much 58 lakhs, madam.
So the grant that we had from Science and Engineering Research Board was 49 lakhs, and that was utilized in-house itself for all the immunological investigations, and all the whole exam and whole genome sequencing were from CSIR IGIB through the Guardian Consortium. Otherwise, we would never ever have been able to do that. So yeah.
Yeah, so the point I'm trying to make is that we are not talking about hundreds of crores to make such difference. Yeah, we are talking about lakhs, tens of lakhs, which can make a huge difference. So the really big important point that we should address is, can we start programs in pilot for genetic disorders which are prevalent? Can we educate the next generation of clinicians to diagnose them and treat them very efficiently? And more importantly, when the sequencing becomes cheap, which is going to become, right? There is no doubt it is going to become. Can we prepare the next generation of workforce which can cope to it and actually become globally competitive, right? And if you think that the world will have doctors to look at genomes, the answer is no. Indians will still have to provide them doctors to look at, right? So can we do this in anticipation, plan it, thinking of a 5-year future and 10-year future is something that we should start.
Thank you, Dr. Vinod. That is exactly what KUHS is planning in with the help of Dr. Geeta.
So
Again,
what's the
from
cost of
Dr.
whole
Sabit
genome
Kumar.
sequencing in Kerala
Yes.
now and where it is done? Okay, very interesting question. Now the cost of genomes is something not something that is constant, because as I said, it is going to reduce every day, right? The cost of consumables of doing a whole genome sequencing, assume that somebody has an equipment, how much resources does it take to sequence a whole genome? It's probably going to cost anything from 50 to 80,000 rupees for one whole genome. Every month it is reducing, and probably the hope is within a decade's time, less than a decade's time, we would have probably a $10 genome, right? Now, the problem is not about sequencing genome. You can, if you have the machine, you have the money, you can sequence genome. But do you know how to analyze and interpret the genome? Is a probably bigger question that we need to answer. And that cost is infinite, right? To create a doctor who can understand a whole genome, the cost of doing that is enormous. So we should not really put the cost of a whole genome, because the cost of the whole genome is as good as people who can analyze it.
One last question. Actually, Dr. Mohandas had already spoken about this. You had talked about doing whole genome sequencing for all newborns. But in our pediatric ICU, there is a subset of newborns and infants where they would have lost one child earlier, similar symptoms at the same age, a positive family history other than siblings. And these children coming and crashing and we being nowhere, and then the child dies before we have a diagnosis, and maybe the third time, fourth time, we may be lucky. So in such situations, is it possible at all to think of having whole genome sequencing in 24 hours?
Yeah, so if you have the equipment and if you have the money to sequence it, it is today possible to do less than 24 hours. Yeah. But the real question is, can we build that capacity in anticipation, right? Of course, it's going to be expensive today, no doubt about it. But the point is, if you don't incur that expense today, we will never be prepared tomorrow, right? Tomorrow it will become cheaper, but then you cannot learn tomorrow, right? When it becomes cheaper, you need to learn today so that you are prepared when it becomes cheaper. So it is possible today. There are hospitals across the world which are doing that. The best example is the Radley Children's Hospital, which offers whole genome sequencing less than 24 hours for all 6 newborns in the hospital. But of course, they are from a different country, different economy, different scale of operations. But we can be smart to do selected single genome sequencing, right? And actually learn the tricks. More important is to actually learn the tricks over time, rather than thinking when it becomes available, I will go back and learn. We should be prepared now.